Hello, this is the Bacteriology 1 tutorial on structure and growth of bacteria presented by the SchrDbb with reference Lippincott illustrated review of microbiology by Cynthia Cornilesson, Fisher, and Harvey. So here we're going to visualize the structure, the growth, and the metabolism of bacteria. So we are going to start with an overview of bacteriology. So you need to know that the cellular wall is divided into two major groups. Is it clear? And that two major groups is made up of eukaryotic and the prokaryotic cells. Now the bacteria make up examples of the prokaryotic cells. Now this is the diagram of a general structure of bacteria. The general structure of the bacteria is inside you're going to have a cytoplasm. Is it clear? And the cytoplasm is going to be enclosed by what is called the plasma membrane. Is it clear? So inside you have a cytoplasm, and here this is the genetic material of the bacteria, which is called the nucleoid, which is made up of a single circular chromosome. Now, just above the cytoplasm, or just above the plasma membrane, we have now what is going to cover it up. It's called the cell wall. Is it clear? And just above the cell wall, we have what is called a capsule, which is going to enclose the cell wall. And now we have pili. The pili are just filaments, membranous filaments, which are going to be used, which are used in order to make the communication of the materials from one bacteria to another. And we also have what is called here the flagella. Is it clear? Now we need to know that there is also what is also called the cilia. The flagella is longer than the cilia, and both of them are used in the movement of the microorganism. Is it clear? So that is the first point that you need to know. Now the next point is this diagram now is going to show you the structure of the peptidoglycan, which is the major polymer of the bacterial cell. You need to know that the peptidoglycan is what actually distinguishes the peptidoglycan is actually the molecule that is going to distinguish a gram-positive from a gram-negative bacteria. So we need to know that you have peptidoglycans or gram-positive bacteria, and we have already seen on the introduction of microbiology how you can distinguish gram-positive from gram-negative bacteria. Now the next thing is this is actually the structure of the peptidoglycan. So now these are the different molecules which make up the peptidoglycan. So in order to visualize the structure of the peptidoglycan, you need to know that actually this is the first molecule here, and this is another molecule here. Is it clear? So it is just made up of a ribose background. On that ribose background, there is this molecule, the NHC C11O CH3, which is going to be placed at this position here. Is it clear? And they are going to be linked by the glycosidic bond, as you can visualize here. Now the sequence of the peptide varies from the bacterial species. So this part here is involved at this part, and now this is the sequence of the peptide linkage here. So you have the peptide bond at one end of the peptidoglycan. Is it clear? Now you need to know that this NAM is also called the N-acetyl-muramic acid, and this NAG is called the N-acetyl glucosamine. So all this and plus the peptide chain is going to produce what is called the peptidoglycan. Now the question that you want to ask me is, why is it called peptidoglycan? It's just called peptidoglycan because it consists both of the peptide and the carbohydrate substrate. Glycan for carbohydrate substrate. Now the carbohydrate substrate is what is you have this N-acetyl-muramic acid, and you have the N-acetyl glucosamine. Is it clear? Why the peptide chain is involved here, and this is the sequence of the peptide chain varies now from one bacterial species to another. Is it clear? So that's what you have to visualize, and this is a sequence of the peptide chain. Sequence of the peptide chains can be visualized here. You have one peptide chain and another peptide chain which are going to do a linkage together, and then you have the N-acetyl-monomic acid binding with the N-acetyl glucosamine, which bind again with an N-acetyl-monomic acid in this sequence. So all this is going to form now what is called the bacterial cell wall. Now you have gram-positive and gram-negative organism. So this is the comparison of the gram-positive and gram-negative cell wall. Is it clear? Now for the gram-positive cell wall, we need to know that there is majorly in the gram-positive cell wall we have peptidoglycan chain, and then underneath the peptidoglycan chain of the cell wall we have the cytoplasmic membrane. Now for the gram-negative, you need to know that what the outer membrane contains the lipopolysaccharides. So you don't have much of the peptidoglycan. The peptidoglycans is only small part here. That's why with the iodine, the iodine is also going to stain the gram-negative. Just that the aldehyde that has been used and the ketone that has been used can easily remove the stain of iodine in the case of the gram-negative. Is it clear? So you see that the gram-negative have a peptidoglycan chain, but the peptidoglycan chain is going to be very small as compared to the gram-positive. So you have the outer membrane here, cell wall. It is majorly made up of lipopolysaccharides. Is it clear? And then you have the periplasmic space in between. You just have a small space of the peptidoglycan as can visualize here. Now this is how the mechanism of the flagella, how the flagella work. The flagella, you need to know that is made up of, there are many filaments inside the bacterial cell cytoplasm. Is it clear? And those filaments is made up of the actin and myosin filament which are attached to those flagella. Now in order for the bacteria to move, the flagella can move in different directions. It can have a rotational motion. It can also have a vertical motion, or you can have a horizontal motion. Is it clear? So all this motion, like Helicobacter pylori is one of the bacteria that move by rotational motion. Other bacteria like Escherichia coli can just move by vertical and horizontal beating of the flagella. Is it clear? And you need to know that inside the flagella there is the flagellin protein, which is mostly involved for the chemotactic movement. And now we need to know that the pili, which is also sometimes called the fimbria, are shorter and thinner than the flagella, and are used in attaching and to make sure that there is a contact between one cell and another. So those are the different things to visualize here. Now this is how spore formation occurs. Now you need to know that there is what is called the reproduction of bacteria. So bacteria can reproduce in two main ways. Can reproduce in an asexual and an asexual way. Sexual reproduction of bacteria just occur when two bacteria fuse together to produce a new bacteria. That's a sexual production. Usually two bacteria fuse together to produce a new bacteria if and only if there is if the bacteria want to share the same chromosomal and they want to have the same chromosomal pattern. Is it clear? But usually bacteria don't do sexual reproduction. It is only in certain asexual that can do such. They mostly do asexual reproduction by mitosis, where the different microorganisms are going to split into two. Is it clear? And one of the form of the asexual reproduction of bacteria is by sporulation. So how does this sporulation occur? So you start by the fact that you have the plasma membrane, you have the nucleus, so it's going to invaginate. So you have invagination of cell membrane to form the four spore. Is it clear? So the four spore is going to be produced. You see this nucleus here is going to be divided into two. Then after the invagination, so you're going to have that. Then the next one now, after the four spore formation, you're going to have engulfment of the four spore. Is it clear? Then you have the formation of a double membrane around the four spore. After the formation of double membrane, you have synthesis of the cortex and the coat an exosporium formation. Is it clear? And then you have the release of the spore. Now when this spore now is released like that, this spore, it has a coat above it which can enable it to resist for extreme conditions. Is it clear? So those are endospore formation. Now these endospore are living inside the cells, so when they are released as such, they can resist extreme conditions like extreme high temperatures and extreme low temperatures. Now this diagram here is going to just to show you the stages of bacterial growth. The stages of bacterial growth is divided into three. Provided that the condition for growth of bacteria has increased, there are three main stages of bacteria. There are four main stages of bacterial growth. The first one is the lag stage. At the beginning, when these conditions start to be better, you see that in the lag phase, what happens is that the endospores are going to break up, and then the bacteria are going to enter now, and they are going to try to grow. The second phase now is the log or the exponential phase. In the log or the exponential phase, what happens is that the bacteria are going to extremely divide into two. They are going to divide multiple times in the log or exponential phase to produce multiple cells. Now when they divide in the log or exponential phase to produce multiple cells, the next phase now is the stationary phase. And then in the stationary phase now, the bacteria now are going to compete with each other because there is not enough food substances in order to sustain again enough bacteria the cell division. So after that, when the bacteria are competing too much again in the stationary phase, it's going to be dead phase where there is not going to be enough food substances to maintain again the bacterial growth. So the bacteria are going to die in the dead phase. So those are the four main stages of bacterial growth in a liquid medium. Now this is the growth of colonies on a solid. So this is the growth of colonies of solid in a nutrient surface. So we start with a single bacterial cell. After a single bacterial cell, now we have the cell. So you need to know that you have time. The number of bacteria after 4 hours is going to increase to 256. After 8 hours, it's going to increase to about this amount. And after that, you're going to see a visible colony of bacteria which can be seen in only 12 hours. It means that bacteria have an exponential growth. It can grow in 2 raised to the power a certain amount based on the time in seconds in the different durations. Basically, now we need to know that this is the overview now of respiration, fermentation, energy production in bacteria. We need to know that there are certain organisms which are aerobic. In aerobic organisms, you're going to visualize what is called fermentation. In an aerobic organism, usually what you are going to have in aerobic organisms, you're going to have respiration, and aerobic organisms, you're going to see fermentation, and you have energy production in bacteria. Now we start with fermentation. In fermentation, actually what happens is that it is mostly fermentation is going to start with the first cycle, which is glycolysis. Is it clear? So in glycolysis, actually the major molecule, you need to know the major molecule of metabolism is actually a carbohydrate, and most of the cases it's glucose. Is it clear? So glucose is going to be broken down by glycolysis. So in the process of glycolysis, it's going to release ATP molecule, and it's going to release NADH molecule. It's going to release ATP molecule, NADH molecule. Is it clear? And at the end of the glycolysis, the final product of glycolysis is going to be pyruvate. Is it clear? When pyruvate is going to be produced, that NAD that was produced in glycolysis is going to bind with the pyruvate to produce either lactate or is going to produce ethanol, depending on the microorganism you're working with. So at the end of fermentation, usually you have lactate or you have ethanol. Usually in bacteria which are resembling like plant cells, they're going to produce ethanol, while in bacteria resembling animal cells, they're going to produce lactate. Is it clear? So that is the fermentation product. Now the next one we have in aerobic respiration. Now is it clear? In aerobic respiration, now in this case you're going to enter what is called the Krebs cycle, and the Krebs cycle is also called the tricitric acid cycle. Is it clear? So in the tricyclic acid cycle, the major element is coming from still from the glycolysis. At the beginning of the aerobic respiration, you see how glycolysis, where the final product is going to be pyruvate. Now that pyruvate is going to enter now the tricyclic acid cycle after being converted to acetyl-CoA. Is it clear? And in the conversion of acetyl-CoA is going to produce N-acetyl NADH. Is it clear? So after the production of NADH, this acetyl-CoA is going to enter the tricyclic acid cycle and produce different molecules. It's going to produce NADH, is going to produce carbon dioxide. And now the different NADH molecules now are going to enter the electron transport chain to produce different molecules of ATP. And now oxygen now and the hydrogen that is going to be released by the NADH is going to produce the water, and the ATP molecule is going to be used now by the bacteria for its metabolism. Now this diagram is going to show you the synthesis of the cell wall. If you know this diagram with heart, you can easily know the different mode of action of the different antibiotics which can be used based on the cell wall inhibition. So you need to know that NAM is going to be N-acetylmuramic acid, NAG is going to be N-acetylglucosamine, BPP is going to be bactoprenol phosphate, and the PPP is going to be cross-linkage of peptide, and DP is diamino pimelic acid. So this, you have already visualized this diagram on the cell wall. Now you need to know that in the formation of the cell wall, it starts with the UDP plus N-acetylmuramic acid, and also UDP plus N-acetylglucosamine. I need to tell you something. You need to know that usually in biochemistry, at the start, you need to know that when you want to do a peptide linkage, when you want to do a linkage to produce a huge molecule, there is usually what is called a terminal head. Is it clear? Usually in biochemistry, when you want to produce a very huge molecule, there is a terminal head, and the terminal head, when you want to produce a cross linkage between carbohydrate molecules, the terminal head is usually the UDP molecule. Is it clear? The terminal head in case of cross-linkage of the glucose molecule is usually the UDP molecule. So you require a UDP molecule in order to make sure that the carbohydrate substrate should become a very huge substrate. Is it clear? So the UDP is going to be bounded to the N-acetylmuramic acid and the N-acetylglucosamine. And now after it being bound, it can bind again to these different amino acids and different molecules to produce the peptide linkage. So you have synthesis of the linkage of the peptide, the pentapeptide blocked by the cycloserine. Is it clear? So now we have the fusion. UDP is going to get out. Now it's going to fuse after the peptide being bound to it. The N-acetylglucosamine is going to bind to the N-acetylmuramic acid, and it's going to bind all that to produce this peptide linkage, as you can visualize here. So to produce the peptidoglycan. So the next diagram now is going to be glycine bridge in peptidoglycans. So this is just going to show you that this glycine bridge and all that is just to tell you that the peptidoglycan is made up of the glycan part and the peptide part. This peptide part is just made up of a linkage of many amino acids, and among those amino acids you have alanine, you have glutamate, you have lysine, you have alanine, you have glycine. The glycine which are bound together like that are going to produce the glycine bridge, and then all that. Is it clear? Now the next thing that you have to note is in this diagram there is what is called L and D. This L represents what is called level rotatory, and this D represents what is called dextrorotatory, and all these are just to tell you the stereoisomeric forms of the different amino acids, because you need to know that level rotatory corresponds to an optical isomerism, which show that it's capable of rotating light, plane polarized light to the left, and dextrorotatory just tell you that it is capable of rotating plane polarized light to the right. So those are the different parts involved on the first part of the tutorial. So from here we say thanks for your kind attention. Don't forget to like and subscribe for our channel.